because I haven't done that yet. There we go. These chemical elements are used and recycled in a variety of ways in the natural environment. And each of these can vary sort of at the subatomic level, where structures like carbon and nitrogen, for example, can have differing numbers of neutrons, which means you can have the same element with two different atomic masses, but the same basic chemical properties. So for example, carbon, there are three well-known isotopes of carbon, carbon 12, 13, and 14. They occur naturally, but in different amounts. And each of those have sort of different purposes in the natural environment. And because it's actively cycled, these isotopic elements vary in their use. And it follows that gradients of their distribution in space will exist depending on the underlying processes associated with the recycling behavior. These gradients are known as isoscapes. And the idea of using isoscapes to determine important areas for marine species, like I said, is not new, goes back to sort of the 90s, and comes from the idea that prey items of marine predators will exhibit isotopic properties associated with their local environment due to uptake through the food web. So the ratios of nitrogen and carbon isotopes can be measured in the tissue samples should theoretically match the ratio of nitrogen and carbon isotopes in sort of the broad oceanographic areas where they're foraging. Most studies examining the relationship between environmental isotopes and distributions are either sort of single species studies or don't really make a whole lot of use of these fine scale GPS data. So the study that I'm talking about here, which is led by Tegan at Nelson Mandela University, made use of both fine-scale GPS data and a multi-species data collection exercise, which aimed to investigate how samples from different species can influence how isoscapes are generated and interpreted, thus acting as sort of a critical assessment of other such studies. The paper does this in the context of three predictions, and I'm just going to read these verbatim from the paper because I think Tegan did a good job of putting these out there. So with regards to, say, the influence of seabird species on the detection of large-scale spatial variability in delta carbon-13 and carbon-15, the paper suggests that they predict that due to vast distances traveled by flying species during a single foraging trip, the carbon-13 and nitrogen-15 tissue values will reflect known baseline latitudinal gradients. However, due to sharp changes in baseline carbon-13 values at frontal zones, we predict that if individuals of a species do not cross different major fronts within the Indian sector of the Southern Ocean, the carbon-13 values of these species will not reflect a spatial gradient. The second prediction reads, we predict that within the isoscapes of species which crossed one or more major fronts, there will be consistent stepwise increases at major fronts from south to north. The third prediction reads, previously it has been shown that an inshore, offshore, and benthic/pelagic effect can be detected in the isotopic values of seabirds. Thus, due to more limited foraging range of penguins compared to that of flying species in this study, we predict that carbon-13 and nitrogen-15 plasma values of penguins will not reflect a latitudinal gradient, but will rather reflect the inshore, offshore, and benthic pelagic foraging habitat of the species. So that was kind of a long-winded way of kind of saying, one, flying species will reflect known isotopic gradients if they cross frontal regions. Two, isoscapes of species which cross fronts will have stepwise increases in isotopic values. And three, isotopic signatures in penguins shouldn't reflect latitudinal gradients, but will represent an inshore-offshore foraging habitats. The authors in this paper tracked 3 albatross species, both species of giant petrel, and 3 penguin species that all breed on Marion Island in the southern Indian Ocean, totaling about 176 individuals for all species. Upon retrieval of the GPS units, blood was taken from the birds and processed accordingly. I'm not going to describe the technical details of this you can have a look through the methods section of the paper. Again, I'm not like an isotope person, so anything I say there would be utterly meaningless coming from my mouth. Bird behavior along the GPS tracks was derived using an unsupervised clustering algorithm. This is basically a type of algorithm that will detect similar behavior types based on parameters in the GPS data, so things like tortuosity and speed. The algorithm they used was called expectation maximum binary clustering. It's actually not one I've heard of before, which is quite, so I'm keen to kind of look at that at some point. Isotopic concentrations in blood have a short half-life, and so to generate isoscapes broadly, what the authors did was take the mean foraging latitude and longitude of each individual. The technical details of sort of how this was derived are available in the paper if you look in the estimation of isoscapes section. And it's an interesting approach. So basically you end up getting, say, a single point for each species that represents where it was foraging on a specific foraging trip. And using that series of points, that map of points, you can apply a spatial Kriging algorithm to determine a sort of a smooth map of the isoscape. The authors found that the values of carbon-13 were correlated to latitude for flying seabirds, and similarly were the values of nitrogen-15. However, there was no significant correlations with longitude. Variation of both the nitrogen and carbon isotopes seemed to be greatest for wandering albatross. There's quite a nice wide spread and a very lovely map that gets generated. And that almost is not surprising. It has the largest foraging range of all the tracked species, and was the one that crossed the major frontal zones more readily. The penguins that were tracked exhibited significant correlations between carbon values and nitrogen values and distance to coastline. However, macaroni penguins, interestingly, which traveled the furthest on foraging trips, did not seem to show much variation in either carbon or nitrogen values when compared to the other two species. So I've obviously not covered, say, every little point of this paper here. So Tegan, to start, I'm wondering if you could touch a little bit on, say, the impacts of diet on this. I mean, you discussed this a little bit in the paper, but is there a way to kind of account for that, those differences in diet? So, you know, if you went out and had a whole bunch of, I don't know, squid, for example, that you sampled, or if you did, say, stomach content analysis and looked at the isotopic signature in the stomach contents, would that help alleviate some of these issues you might be thinking are caused by differences in diet?
I definitely think so. You could get some stomach samples and try. I haven't looked too much into Bayesian mixing models to try to figure out exactly where the signatures in the isotopes are coming from. But once you get an idea and what is exactly in the diet, you can start to see or start to infer where the differences are coming. So we don't really have any recent diet from these species on Marion Island. So a lot of our interpretations in the discussion was taken from previous studies, which was mostly in the 1980s and 1990s. So we're not sure if the diet has changed up until now. When I was working with the birds in Marion, I did see significantly more carrion in a lot of the albatross diets. So yeah, we would have to look into that. But yeah, it would help.
Yeah, it is an interesting. I mean, I guess, like I said, I'm not an isotope person, but I wonder do different species integrate carbon and nitrogen isotopes at different ratios compared to, say, you know, will fish have different CN proportions versus, say, squid, or maybe even top zooplankton, top level zooplankton?
No, definitely. As you go through the food chain or the trophic web, the carbon gets fractionated differently along the way, and different prey have different lipids as well, which changes the CN ratio of the isotope sample.
All right. Okay. So the other kind of question I had was around, say, foraging depth. So you're tracking penguins here, and the different species of penguins will forage around at different depths. And will those foraging depths potentially impact the CN ratios that you're seeing in your data?
It's very difficult to say. So there has been a study done on tuna, but tuna dive a hell of a lot deeper than penguins do. The deepest diving species here is the gentoo, which is 200 meters. I think the biggest impact would just be the prey species themselves. So if the prey are eating benthically at the bottom, they will naturally have a higher CN ratio. But whether the diving of the penguin affects the CN ratio, I don't think it would have that massive of effect, just because they don't dive that deeply. It's more about the prey, whether the prey are found at the surface or found along the bottom.
Yeah. Okay. The other one I have for you as well is around, say, the impact of fishing discards. Now, I know probably I guess probably in your part of the ocean, say for some of those species, you may not be getting a whole lot of fishing vessels, but say for, let's say, giant petrels and wandering albatross, which are highly attracted to fishing vessels, do you think there'd be any impact of, say, fishing vessels bringing down bait types that wouldn't be reflective of the local CN ratios? If you have a factory trawler or something like that comes down and has a factory longline vessel that has stored bait that they brought from up north. I mean, this might be kind of grasping at straws to try and look at unusual factors that might impact these CN ratios. But did you see anything like that, or was there any indication of something like that in what you did?
That's a very interesting question. Definitely, because yesterday I had a discussion about illegal dumping before they come into the harbors. So it could be very likely that they're fishing somewhere else in the ocean, and as they get closer to South Africa, they dump a whole load of biomass that they're not allowed. That's above their total allowable catch. So I didn't really see any of that, just because the wandering albatross gradient was so strong, and they're the ones that would most likely be eating fishery discards. Yeah, I don't know how much fishery discards they would be eating that it would actually change their isotope signature. It would be something to look at. What I brought up in the discussion, that the two giant petrel isoscapes didn't correlate so nicely. They didn't have this very strong latitudinal gradient. And there I said it's most likely because they're eating carrion of birds that forage somewhere else and have been washed into the current or into the fronts, and their signature is different from what would be expected in that area. Yeah, it's a very interesting question, and yeah, I think it shows that we should be using caution when interpreting these isotope values we're getting from the blood of these predators, just because we don't know where their prey is coming from. Mm-hmm.
Yeah, I mean, you do see that quite a lot. I think that's often the criticism of most of these isotope papers, is that there's not enough information on the prey types to really make reasonable assessments of what what they're actually doing at sea, or certainly using the isotopic values for inferring distribution is often criticized for those sorts of things. David, I'll hand it over to you for a couple questions if you've got some.
Yes, sure. Thanks. And Yeah, this morning we have two papers, and in one paper, 175 birds were studied, and the other one 176. So so you can clearly see that we're now in the age of massive deployments of tracking devices, and in this case linked with isotopes. And congratulations, Tegan, on the really massive study. Really interesting and pretty massive paper also to read. And while reading it, I was thinking that you have this luck in the Southern Ocean, so to say, that things are relatively well structured in terms of isoscapes, because the way the Southern Ocean is made with different fronts nicely aligned along a latitudinal gradient. So so that's probably a tremendous help for this type of study in the Southern Ocean. And I was wondering if you had any clues on how applicable your method might be in other areas where things are less structured.
Yeah, that's very interesting. So I'm just thinking of a study that was done, I think, in the North Sea with jellyfish, and it reflected quite well because their tissues, their half-life is quite short, so they could pick up these very quick turnovers of the baseline isoscape. The method I used with the birds, so then, as you say, the nice thing about the Southern Ocean is it's very structured, and you lose that temporal variability at the base of the food web. If you did it somewhere else, you'll probably get, say, in the North Sea that is so variable, or one of these massive currents, you'll probably get the more stable long-term gradient. But as I suggest at the end of the paper, is that you need to start introducing a temporal aspect into this type of method. So you need to maybe do multiple tracks with multiple blood samples so that you can try capture some of that and understand if the method is actually working and is actually viable to use across different isoscapes.
And I was trying to figure if you would move into an unknown area with very few studies on seabirds, and you had, for instance, a small species of endemic petrel you cannot track with devices, and but you would have very little previous knowledge of the marine environment. What would be then the first logical steps to try and use a stable isotopic analysis to infer where they've been to? For people who would want to transfer this type of methodology to areas with far less knowledge than the Southern Ocean.
Yeah, it's a tricky question. If you can't track the animals, I guess you'd have to do sea-based observations and get an idea of where they're foraging, and try model whether they're going after an oceanographic feature or some high productivity oceanographic feature, and then try see if there's variability in their carbon, if the carbon is actually reflecting anything in the environment. So maybe perhaps there's sexual segregation, and maybe you could identify the sexes at sea, which would be lovely, but not always possible. And then you could try see, okay, can we link the variability in the isotopes to where we're seeing the two different sexes? But it would be quite difficult. I would try maybe a baseline study, so getting the organisms at the baseline to try to see if there's spatial gradients, or maybe something larger that's a bit easier to work on, even if the birds aren't preying on it, something like cephalopod or fish or jellyfish or something like that, that you can start to get a pattern.
But if you're like in faraway places of the Southern Pacific, then probably it would be easier to try and actually track the birds than to first infer isoscapes to try and see where they've been to.
Yeah.
And I see in the comments that Virginia put in something about the Mediterranean. I was wondering whether Virginia would like to give some insights about the situation in the Western Mediterranean, whether because I know the Barcelona group has been using stable isotopic analysis a lot. Whether Virginia wants to make a comment about how this works or does not work in the Mediterranean.
Will I unmute you, Virginia? Give me a nod if you're all right. Yeah, okay. I'll unmute you. There you go. You're unmuted.
Thanks. There's construction around, so if there's too much sound, just let me know. We've been working for a while with stable isotopes, but we've been using them more for diet reconstruction than for isoscapes and geographical assignation. I know there's some people in my group that did that a while ago. And what it helped quite a lot also was to collect dead birds by caught on longliners, for example, and then use their stable isotopes in their feathers to try and infer that. But if you have a species that you cannot track, probably what Tegan was suggesting about using baseline data and then trying to infer how that would change into higher levels. And so it would be tricky because there's a bunch of other stuff that would affect the signatures of predators.
In the Mediterranean, you would probably get some sort of binary information on whether the species is a lot on the shelf of the Gulf of Lion or farther offshore, more in blue waters, right?
Yeah, shelf offshore is a pretty clear signal. Although I think there's a region, I think, in the Gulf of Lion that has a very weird isotopic signature that seems almost Atlantic. But yeah, also the isoscape, the baseline isoscape in the Mediterranean is pretty well known as of today. And I don't know if there's a region where the isoscape is not as well known. I guess that's the first step you should take. But yeah, using carbon mainly, platform offshore is using carbon is pretty easy easy, not complicated to differentiate.
Yeah, thanks.
Thanks.
You're welcome.
Yeah, well, Grant, that would be all from my side.
Okay, great. Well, that's perfect timing because we've got a question from Eric Voller. Hang on, Eric, let me unmute you. I'm trying to unmute you. There you go.
Okay.
You're good.
Terrific. Tegan, it's a great paper. I really liked it because one of my criticisms of past isotope papers has been the lack of independent verification of foraging areas. Studies would take samples of blood or feathers or whatever and then make inferences about where the birds were foraging based on the delta ratios. So having the GPS data to independently verify is a really sweet addition to the paper. So I really congratulate you on that. The question that came to me is, why didn't you play with king penguins? Because they will be a species that will be halfway between the flying birds and the near-shore shallow water penguins. We know from Hurd, Kerguelen and Crozet that the kings will travel all the way down into the ice zone. So that would have given you a species that would have crossed multiple oceanic fronts and gone into very different oceanic waters. So it's not a criticism, I'm just curious, was it just the fact that you didn't want to play with kings through the winter time or what?
So I'm glad you brought that up. It was actually a great pity. We did try get data from the kings, but all the GPSs got flooded on the way back. So we only had about 4 individuals, so they didn't make it into the paper.
Right.
Shame.
So is that because the birds dive too deep for the units or what?
I think so, yeah. Yeah, I don't know what they were up to, but they managed to break them all.
Yeah. And so no results whatsoever from kings?
We have, I think we had about 4 or 5 individuals, and their isotope actually, they all went, so the Marion king penguins go straight towards the Antarctic polar front, and their signals matched the wandering albatross, which was, it would have been great to add them.
Yeah.
But so now recently, since I finished my data collection, Pierre Pistorius, my supervisor, has continued collecting, so we can maybe do a short note just on the king penguin isoscape and add it in.
Yeah. As I said, it wasn't a criticism. I was just curious because to me it was like, kings are such an obvious contrast to the other three species that you worked with.
Yeah.
It would have been great, but unfortunately we didn't get it.
No, all good.
Thanks, Eric. Tegan, we got a question from Andre. He said he's asked who made the GPS units.
I don't know if I should say this if it's going on YouTube. It was.
You could probably message him privately, and it won't come up
Okay.
on YouTube. There you go, Andre. I hope that'll answer your question without incriminating poor Tegan. Do we have any more questions from the audience? I don't see anybody flailing hands or raising hands on the screen, so I think with that, we'll probably move on to the next paper. So Tegan, thank you very much for joining us. Thanks for the really cool paper. Hope you stay tuned for this next little discussion we've got.
Yeah, of course.
Penguins.
Thanks, guys. Yeah, thank you, everyone. And thanks for the interest in the paper.
Thanks for joining, Tegan.
It's a pleasure.
All right, we've got a penguin paper coming up. I love penguins. This is great. So we've got Yuki and we've got Akinori. Hello, Aki. Hello, Yuki. How are you?
Hello, I'm fine.
Nice to see you both.
Nice to see you.
Nice to see you again.
Nice to see you again.
Very long time. Very long time. Yeah. Yeah, you both look exactly the same. Incredible.
You look the same too.
No, that's not true. That's a lie.
Yeah. Don't stroke his ego too much. He doesn't want to feel like he looks that young.
No. That's too polite. Well, thanks for joining, and we should kick off with the second paper. And what I have to say is that a bit more than 10 years ago, Emerson and Southwell wrote in the journal Ecology, I quote, "The relationship between Adelie penguin and ice is well established." As so often in science, things are not that simple, and there's still considerable debate about this point. Adelie penguins are, together with the snow petrels, Antarctic petrels, and emperor penguins, iconic sea ice obligates, and consequently, everyone would like to know what will happen to them in the context of global warming. To the general public, the matter is simple. Sea ice is vanishing, and with it the Adelie penguin, just as polar bears on the other side of the globe. Well, it's really not that straightforward because firstly, at the moment, areas of the Antarctic Peninsula are losing sea ice, but on the other side of Antarctica, for instance, in the Ross Sea, there is more sea ice, and for other areas of continental Antarctica, things are relatively stable despite strong interannual variation. You correct me, Yuki, if I tell nonsense. Second, there are many different types of sea ice, and important for Adelie penguins, they're land-fast sea ice or grounded mega icebergs, which may prevent them from accessing the open sea. But fast ice also hosts zooplankton, especially krill, on its underside, and this is accessible to the penguins through cracks. Yet what the penguins obviously prefer is to forage a bit farther offshore in the marginal ice zone, where sea ice concentrations of about 20% seem to be optimal for them. But there's also a lot of debate about that. So the scientific debate is raging at the moment, with teams working in different areas of Antarctica, finding contrasting results. And in between them, in the crossfire, there are modelers trying to infer future Adelie penguin population trajectories. And the poor modelers, they're getting restless because people in the field keep on telling them different stories on how penguins react to sea ice. So very complex system. And it's in this context that appears a high-profile paper led by Yuki Watanabe from the Japanese National Institute of Polar Research in Tokyo and his colleagues. And Associate Professor Watanabe has a very long experience together with his colleague Takahashi of working with Adelie penguins, and they kicked out a long series of very nice papers on the penguins. For this publication, the team worked at Lutzeholm Bay in Droning Maud Land at the Japanese station, facing the southern Indian Ocean. They equipped the birds' breeding adults with GPS trackers, accelerometers, some of them on the body, others on the head, so that they could infer prey capture events, and sometimes cameras. And they detailed the foraging effort and the foraging yield of the birds. And for a subset of the birds, they also measured body condition, diet, and chick growth rates, breeding success. They did this for 4 years across the last decade, 3 of which with a lot of sea ice in front of the penguin colony. There's a picture in the paper showing this. And the 4th, during the 2016-2017 breeding season, the 4th water was completely open in front of the colony. So a big change. And obviously that changed the penguins' behavior a lot. Because when there was sea ice, they had to walk on it for a long period before diving for krill and fish in the cracks. Diving in those cracks was also prolonged the ascending phases of the dives, probably because they had to spend time finding the gaps in the ice to get back to the surface. When there was open water, the penguins had shorter foraging trips, yet they traveled farther, and that's because penguins are so much faster when swimming than walking. And their dives were shorter and they yielded more prey per unit time. As seen on the camera recordings, the water around them turned from blue to green because in the absence of sea ice, the productivity of the whole coastal ecosystem was boosted to the benefit of krill swarms. Consequently, because foraging was so much better, so was also adult body condition, chick growth rates, and the breeding success. They were all higher in the year without sea ice. So overall, the authors come to the counterintuitive result that no sea ice is good for Adelie penguins. So I had several thoughts about this paper, and the first was that the study recalled the conclusions of the 2002 paper in Polar Biology by Akiko Kato and colleagues, which is quoted now in the Science Advance paper. And in that paper led by Akiko Kato, the authors studied long-term population dynamics at this site, and they concluded, I quote, populations tended to increase during or after periods of sparse sea ice in summer, a condition that occurred once every decade. So it seems that this open water condition is pretty exceptional and happens once in a while. And then things started to make a bit more sense to me when I bumped into a paper published this year in Global Change Biology, and it's a paper led by David Iles, I-L-E-S, sorry if I mispronounce it, from Woods Hole, and it's a collaboration with the Shise Lab. And in the paper, the authors say, I quote again, sea ice predicts long-term trends in Adelie penguin population growth, but not annual fluctuations. Populations tended to increase during or after periods of sparse sea ice in summer, a condition that occurred only once every decade. So the way I read this is that, in a sense, your study showed a momentary positive effect of sea ice disappearance, but in the longer run, less sea ice may be indeed challenging for Adelie penguins. Is that right? And then if this is right, what sort of conditions influence the penguins in the longer run, and maybe also through the interbreeding phase?
Yes, I think the balance is the most important in we call ice-free season, because we have no sea ice in front of the penguin colony, but this is kind of perennial. So we have still sea ice offshore, which means they are protected by, for example, leopard seals. They cannot come into the penguin colony. So we say ice-free season, but this is not complete. I mean, this is not completely ice-free for all that region. So what we the situation what we saw is kind of ideal situation, still surrounded by sea ice, but penguin are free to dive and swim.
Okay, and so offshore there's still sea ice, and what sort of concentration of sea ice was there offshore in that particular year?
Sea ice was thin, but still the bay is covered by sea ice.
Ah, okay, so it was a complete cover outside of that polynya.
Yes,
so,
yes.
Okay, so basically they were feeding in a highly productive polynya just in front of the colony, and there was the additional benefit of being protected from the leopard seals. And so what do you think? Is there still a possibility to give out a general message about the impact of sea ice on Adelie penguins for the whole of Antarctica, or are the regional differences so strong that it's not possible to give a general message?
I think the contrast between Antarctic Peninsula area and continental area is quite strong. And in continental area, including our study area, there is quite clear tendency that penguin population start growing when sea ice was very sparse. So I think that signal is very clear.
Okay, and since we're talking about the Antarctic Peninsula, I think that Grant had elements to provide about this point.
Yeah, it's quite interesting when I saw this paper. This is actually for the Aki, you probably know Ron Naveen, of course, and Ron was the one who shared this with me there a couple of weeks ago to discuss, because obviously it's quite a different story than what you get in the Antarctic Peninsula. And when you talk to advocacy groups and such, of course, they always discuss how climate change is reducing sea ice, and therefore penguins are all going to die. And this kind of runs counter to that narrative, and that's very important and interesting because, and oftentimes when we talk about penguins with passengers on cruise ships, the one thing that we try to bring up is the fact that Adelie penguins south of the Antarctic Circle do something very different from penguins on the Antarctic Peninsula. We always attribute that to climate change, but we really don't have the strong enough evidence for that area yet to be able to make that conclusion. So in what you guys find is actually intuitively makes lots of sense. Energetically, a penguin to walk is challenging, and a lot of this stuff sort of harkens back to the work that David Ainley did in the early 2000s, where he was looking at polynyas and such in the Ross Sea. And when you look at population trajectories of Adelie penguins in the Ross Sea, as very comparable to what you guys are seeing in East Antarctica, where populations of Adelie penguins are either stable or increasing. So overall, over the entire peninsula, or the entire continent, Adelie penguin populations are actually either stable or increasing, just because of the huge numbers of Adelies down south of the Antarctic Circle. I'm wondering, so do you believe that, because you're looking at sort of like an event, it's a single event that's had a positive influence on the population. Do you think that, say, on the Antarctic Peninsula, part of the issue might be sea ice has been gone for so long that it's influenced krill dynamics in that area? And do you have any comments on that, for example, or any ideas as to what may be influencing populations there?
Yes, I think it's possible. sea ice can facilitate krill production. So it is possible for penguin in penguin Antarctic Peninsula regions.
Mhm.
Yeah, that really makes sense. I was thinking the same while reading the paper. I thought, well, you have this sort of lid on the ocean with the sea ice, and once in the decade it vanishes, and then of course you have this huge bloom of productivity. But if this would occur year after year, maybe it would not remain as profitable.
Mm-hmm.
Yeah.
So can I add to that? So I think, yeah, our study area, there are lots of sea ice, in usual years, and we had 1 year of like a middle level of breakup, and so there's still some ice, but physical barriers for penguins to dive is removed. So that's kind of ideal situation for Adelie penguins, and I think. Yes, so looking overall, Antarctic areas, what we see is probably, too much ice is no good for penguins, but too less sea ice is not good for Adelie. So they need some level of sea ice to ice algae to sustain krill populations, but they don't want to, too much ice to prevent them going to dive into the sea. So I think our study area is looking at the like a heavier sea ice side of the entire environmental spectrum, and in the peninsula area there's much fewer sea ice areas. So kind of there should be some optimal sea ice concentration, and yeah, East Antarctica is like a heavier side of the sea ice for the optimal condition, and peninsula area should be the smaller sea ice extent than optimal. So that's my understanding.
Yeah, and since we're talking about the different areas of Antarctica, while reading the paper, I couldn't help thinking, what would Dave Ainley think about the situation and the conclusions? He's known for his very strong opinions on the matter. So
yeah, we actually had that as well. So the moment that David found out that we were having this discussion, he sent me an email, and we had a few back and forths about this very topic, because he was very interested. He does send his regrets that he can't be here today. It's far too early in the morning for him, but I'm sure he'll watch later and catch up with the discussion. But yeah, I mean, this is just it. First, David would have lots to say about this, and I think he very much agrees with the interpretation of the results here, that these birds they love these polynyas, they love these little cracks that they don't have to walk far to get to. Vastly increases their energetic efficiency, and that's leads to some interesting discussions around what's going to happen in the future with penguin populations as sea ice really starts to decrease around the continent, which of course we haven't seen massive impacts on yet, but it's clearly it seems to be coming. So actually that might be a question for you guys as well, is do you see there is some evidence that sea ice is decreasing around the continent now, particularly in East Antarctica. Have you guys seen evidence of that in around your station, or is that not evident yet?
So our so the year the we call ice-free year, which is from 2016 to 2017, and this is the year when the overall sea ice extent in Antarctica start declining very quickly. So actually we observed, we thought this is a very local phenomena, but after that we knew that this is not local, kind of a more big scale event.
Interesting, yeah. Okay, I think we've got a question from Eric again. Eric, I'm just going to unmute you. Hang on a second. There we go. Go for it, Eric.
Okay. Yeah, just Grant, going on to one of your comments. I was just going to throw in a comment that would have come from Dave Adelie. we've seen a lot of discussion in the literature, say in the last 20, 30 years, dealing with the role of sea ice and this sea ice optimum that you mentioned as well, Akinori, in terms of the Goldilocks, not too much ice, not insufficient ice, but somewhere in between. There's also the other question about the recovery of the whale populations now starting to take large amounts of krill that we believe perhaps during the 50s, perhaps to the 80s or so, the penguins were taking advantage of a decrease in the whale population. So there was this so-called krill surplus. So disentangling all these various threads is always challenging. And the nice thing with the Adelie penguins is that we have them at 3 broad latitudinal ranges. We've got the northern populations on the peninsula. We have a significant proportion of the population around the main body of the Antarctic continent at around about the same latitude, give or take. And then you've got this wonderful chunk of the Antarctic that is the Ross Sea that goes 10 degrees farther south that has a different sea ice regime. So you actually have the potential for doing some holistic whole of Antarctic type analyses, looking at regional whale populations, regional sea ice extent, and regional trends, because we've got relatively robust population data. I mean, that's the project from hell, but it would be a fun one to put together those disparate data sets and to see if there are some common threads to the various populations trends that we're seeing, so that we get a better understanding or insight. Mm-hmm. So again, I think this is a really good paper. I enjoyed reading this paper as well. But there's always that, how can you translate the results from one study to the entire population of a species?
Mm-hmm. Yeah, thank you. I think this will be a great project. I found that especially the combination of GPS and accelerometer is very strong, because we can know where they go, and we can know what they did, whether they are walking, swimming, or diving, or resting. So the combination of GPS and accelerometer will be, and attaching those devices to penguin in whole Antarctic would be a great project.
And since you're mentioning that part of the technology, so in this paper you used accelerometry data to infer prey capture rates and mm-hmm. not the camera data. Mm-hmm. What were the reasons for this?
Because one reason is we deploy camera on part of the penguin, not all animal, and another reason is especially in ice-free season, penguin dive to greater depths. So often the image was very dark. It's difficult to see what was happening.
Okay, so probably the camera data are good for ground truthing some of the accelerometry data, and yes. Also more for qualitative qualitative analysis and not necessarily quantitative.
Mm-hmm. Mm-hmm. Yeah. So yes, camera is very beautiful, but it's kind often difficult because the image is too dark or cannot see, or it can happen.
Yeah. Yeah, there's some, I think it's the folks from the Norwegian Polar Institute are using cameras on chin straps at the moment, and getting some really cool imagery. But yeah, they're reporting the same sort of issues, that once the animals get to depth, the cameras are basically useless. But you do see some very interesting, you do get some interesting sort of like conspecific fishing and foraging that you pick up on the cameras, which is really fascinating. Andre says down in the chat that he thinks the camera only lasts for a couple of hours. What's the battery life on these cameras?
Camera battery life?
Yeah.
It was 2 hours, 3 hours, something like that. Not so long.
Yeah. Yeah. Okay. I assume that the cold probably impacts battery life as well. Mm-hmm. Yeah. Yeah. Let's see. Do we have any questions from the audience? I'm not seeing any hands up.
Maybe before you move on, I just want to add some comments on like our study situation.
Absolutely.
So yes, I guess Grant, you see many Adelie penguins colonies, but in our study areas, the population size is very low. So our study colony is just like 150 pairs or so.
Right.
So we have small colonies. That is partly, I think, due to the heavy sea ice conditions. So in normal heavy ice years, the population of penguins are regulated by small availability of foraging site. Then when they have open sea ice situations, they can enjoy per capita foraging success. But if those like open ice conditions continues, and if penguin population starts to grow, then you would see massive Adelie penguin colonies, and they may not have enough food. So in our situation, I think the density dependent population regulation is like removed or something by sea ice, but maybe not in other colonies. And also I'd like to point out that sea ice, heavy sea ice had bad effect on penguins. That is also recorded recently in French Devon station. So Jan Loperclel and colleagues, they recorded the almost complete breeding failure of Adelie penguins in years with like a fast sea ice extend much from the coast. So I think that the fact that the sea ice act as an important physical barrier for Adelie penguin is not only in our location, but it can be applicable to various continental Antarctic study sites.
Yeah.
And to elaborate on Eric's question about competition with marine mammals, apart from leopard seals, do you have continuous presence of marine mammals offshore, which may compete with the penguins, or because of the thick sea ice situation, it's not that much of an issue in normal years?
I think. So yes, in our areas, the penguin doesn't go far. So like they stay within fast ice areas. So whales cannot come into the Lisbon Bay, and only like weather seals are feeding along the kirk. And so there's no competition as far as we could see.
Do you get killer whales?
No, maybe much further offshore. But
okay.
Not in Albatross.
Okay.
That's quite cool. And going back a little bit to your point about sea ice acting as a barrier, you could almost see how in places like the Antarctic Peninsula, how you might get sort of a population blip after the sea ice disappeared. So if once you got to a point where the sea ice was gone completely, there may have been an immediate increase in the number of birds that were breeding there. And interestingly, this past season, I had a passenger on board the ship I was on who shared with me an image from Paulette Island from the early mid 2000s. And the picture shows an area when you land on the island that's completely barren of Adelie penguins. But if you go to Paulette Island now, Paulette Island is almost completely covered with Adelie penguins in that area that was in that picture. And they were taken about the same time of year as well. So it's not like it was a timing of breeding issue. But yeah, certainly Paulette was seemingly had a lot more birds on it than it did in the 2000s. And interestingly, Paulette is an island that we don't have a lot of data from. We recently took a drone survey of that island. So it'll be interesting to see what's happened there. But yeah, there's this kind of idea that there might be an initial population blip and then a decline again. So it'll be, of course, it's always interesting to follow these things through time as we're doing these long-term monitoring programs. Eric has another question. I'll unmute you again there, Eric.
Yeah.
There you go.
Grant, I'm just going to remind you that Adelie penguins typically don't start breeding till they're about 4 or 5 years of age, and so that blip may not happen until you see that recruitment back into the population rather than the next year.
Yeah. Yeah, absolutely right. And so these initial blips might be time lagged, and you probably, you know, if it occurred with other species, of course, you might see it there as well. I guess before we go, I might just ask one more. Oh, actually, Yutaka has a question. Hello, Yutaka. I'm just going to unmute you here. Hello, Yutaka. How are you?
I'm fine. I'm fine.
Nice to see you.
Talk and story. And I have a question. I understand that the krill and sea ice are the key factor controlling the foraging and population of Adelie penguins. And at this moment, Antarctic krill or ice krill are only available prey for Adelie penguins. But in the sometimes later, the other prey can be available. And my question is that can the Adelie penguins feed on switch to the other prey species?
Yes, I
can do, but I'm wondering if the Adelie can do so or not.
My understanding is yes, they also eat fishes, especially under ice fishes. So we got a lot of video image showing penguin capture fish just underneath the ice. But looking at our long-term data, the year when penguin eat more fish have lower breeding success. So it looks like yes, they can switch to fish, but it's not the best option for them. This is my understanding.
Yeah, and that's sort of how we interpret it as well. Is that Adelie penguins typically are referred to as krill obligate feeders, but they can certainly eat fish. It's just not their preferred prey.
But I think what Yutaka also probably meant is it possible that completely different species of fish might move into this system?
Yes, yes, that's true.
That's probably the point. And then you know, we've seen that in other systems that then seabirds can be surprising in their flexible diet. And even though, you know, we so strongly associate Adelie penguins to krill, you know, there might be a third player coming into this game in terms of prey type, and we might be all surprised. Just needs to be nice and fatty.
Yes.
After the sea ice completely disappear, the other prey can be available in the shelf slope area where the Adelie penguin can reach.
Yeah, that's another decade of research, Yutaka.
Yeah, that's true. I'm too old.
Interesting fact is we in the ice-free year in ice-free year we observed quite a lot of penguin eat a lot of jellyfish at that year. But how I'm not, but I cannot quantify how important they are. But we observe jellyfish eating event many times.
I think in Rossi, yes, I think about half of the prey like Antarctic silverfish, not krill. So I think, right. I guess, yes, David will be yes, embarrassed. We always say, you know, the Adelie is a krill obligate, but that's not the case in Rossi. And I think, yes, so like a fat and, you know, abundant fish like Antarctic silverfish, if those come into the ecosystem, then Adelie may be able to be sustained on that. But yes, so we don't know, you know, fat species will become available after sea ice is gone. So
yeah, it's an interesting question, and yeah, like David said, another 10 years of research. So for those of you who are young enough to still be around in 10 years, good luck. All right, everyone. I don't see any more burning questions, and so I think with that we'll probably close it off because it is 20 to 9 here in the UK, and I'm sure most of you want to get on with your days. Thank you, Yuki. Thank you, Akinori, for joining us. Thank you for such a cool paper. That was great fun to read, and I'm sure us in the penguin community are going to be discussing that for a while, and how we're going to interpret that with the growing number of passengers going to Antarctica. We're all asking, how are penguins doing in Antarctica? And David, of course, thank you as always. And
it was great to see everyone.
Yeah.
But thanks so much for joining.
Yep. Thank you, everyone. And yes, we'll see you guys again, hopefully next week or the week after. Next week, we're back to 1600 GMT, where we'll have a couple other papers. And then with that, I'll let you all go. Thank you very much. Stay safe, stay healthy, wash your hands, keep masks on. Take care, everyone.
Bye-bye.
Bye now.
Bye.
bye.